CSIR Fellowship, DST Young Scientist Award he achieved, and he has 50 plus research publications and 30 plus paper presentation at international level or national level. He receives award from Society of Toxicology, Therapeutic Drug Monitoring,
CSIR
and
Fellowship,
AMP,
DST Young
Clinical
Scientist Award
Toxicology,
he
Central
achieved,
Nervous System,
and he has 50
Cardiovascular
plus.
System. So, we enlightening the profile of Dr. Ravinesh Mishra. I would like to invite Professor Dr. Ravinesh Mishra over to Professor Dr. Ravinesh Mishra. Please, sir.
Thank you, Mr. Sunil Dutt, for a introduction. Firstly, I would like to
Thank you, sir.
congratulate all the organizers and their team for inviting me at a international platform, that is International Online Conference of Drug Discovery and Lead Optimizations. Today I will speak on role of impurity profiling in drug discovery. Is my presentation is visible to the audience?
Yes, sir. Yes, sir.
Okay. So prior to the start of this impurity profiling in drug development, I would like to enunciate about the discovery of the drugs which have been discovered for their purpose, but due to their side effects or due to the substances which interfere with their exactly to be produced their therapeutic effect, they have been used for another kind of the therapist, means another kind of the treatment, like minoxidil. So minoxidil is a drug which is basically used for the treatment of hypertension. But the side effect of minoxidil after the post-market surveillance of the clinical trial phase studies, it was observed that this one is having some problem with related to the growth of hairs on the body. Even on the palm, if you are going to use excess doses of this minoxidil, that leads to the growth of hairs. When these studies or TDM monitoring processes, the clinical data went in the market, then again back to the lab, then scientists, researcher, and they develop means oral suspension or the suspension which we can apply on our topical skins and used for the means for the growing hairs, and that is a very effective drug topically in the baldness or in the other kind of related hair problems. So that is impurity profiling, and most of the drugs have been discovered, but in India, means nowadays the scientists used to work on the impurity profiling, but this is the area where our scientists can focus and develop new molecules which are going to have a higher therapeutic value, or it may be without side effects. So what are the impurity? So impurity in pharmaceutical basically. So I am going to discuss on the impurities in pharmaceuticals. So impurities in pharmaceuticals are the unwanted chemical that remain with the active pharmaceutical ingredients or developed during formulations. Because these APIs used to prepare by the types of the reactions, So some traces may present in their end products, which we may or may not enable to separate from our end products. That's why they result in our formulations, or they may exist during the preparation of these APIs. The presence of these unwanted chemicals, even in small amounts, may influence the efficacy and safety of the pharmaceutical products. As we all know that there are some potent medicines which are effective in very, very small doses or in minimum concentrations. So in that case, if there are presence of any means undesirable components with medicines, that is going to affect our body, so there is a need to control impurity. And what is the why we are supposed to do this one? So this one is the necessary to ensure the safety and efficacy of pharmaceutical products. Even we are required to determine the purity of the drug product. So, what are the various types of impurities and their sources? So, impurities like process related drug substances, they may be kind of organic or maybe starting materials or the intermediates. Then, process related drug product, organic or it may be inorganic, then reagents, catalyst, etc. Because we are using number of ingredients or reagents or the equipments for preparation of APIs or any drug formulation of finished drug product, even during degradation of drug substances. So it may be kind of a organic product or degradation of some chemicals. For example, we can see the normal like benzaldehyde solution which are we are using in our daily lab. So if there is a kind of oxidation formation of benzoic acid crystal over there, so we don't know when these kind of reaction may takes place, and their form may change also, the end substance also. Then degradation drug product, it may be then excipients interaction also, because while we are going to prepare any finished formulation, it may be like capsule or it may be in injections or topical applications. So we should supposed to use excipients. Even to mask the taste, we use some excipients. So there is some compatibility with excipients and APIs. So there are means various materials used for the preparation of any drug in a kind of a patient compatible form. So, sources of impurities in formulations. So, as I discussed in the previous slides, so impurities may be associated with the active pharmaceutical ingredients. It may be organic impurities, it may be inorganic impurities, it may be residual solvents, like solvents we are using for these reactions. Then, impurities created during formulation, it may be process related, it may be environmental related, because from a like a manufacturing place the drug used to be transported to the different temperature regions, it may be some tropical or in other countries too. Then dosage form related, then functional group related. So considering all these points, there is a like accelerated stability testing nowadays used to conduct to check the shelf life of any finished drug product. So these are the some means sources by which we can identify the impurities in formulations, and there are some protocols already have been designed to identify the impurities found by during above these mentioned processes. Then categories of impurities: organic, inorganic, residual solvents. So organic impurities, these are arise from the manufacturing process and or during storage. So these are maybe classified like those are identified impurities, those who have some structure has been determined, and there is some value have been fixed by the specified books followed by the various industries of and like if there is a zero presence of 0.2%, if the toxicological data is available, if that value is less than 0.2, that is 0.1, if no toxicological data is available, and some may be unidentified organic impurities, those whose structure and toxicological are known and their limit is 0.1%. Then organic impurities includes means it may be starting materials, it may be byproducts formed during the reaction, then intermediates, then degradation, or even maybe with finished products also, then reagents, ligands, catalyst. Nowadays we are also using some like microwave radiation techniques for the preparation of these finished products. So in that case, some carcinogenic rays may be also may some possible changes which may not able to identify our analytical methods. Then identification of impurities. It is based on the chemical reaction involved in the senses, any raw materials used which could contribute impurities or any possible degradation product. So we should supposed to have a concept of understanding of the chemical reaction, reaction conditions, and any metabolic degradation pathways is essential for identification because there is a like a phase 1 metabolism and phase 2 metabolism for the degradation of our drugs. So there is a drug which belong to the category of diuretic, that is hydrochlorothiazide. So hydrochlorothiazide degrades to the sulfonamides. That is a means basic metabolic pathway of this hydrochlorothiazide. So we should suppose to ensure the acceptance level of this disulfonamide within body. So a specific impurity test is conducted to determine the amount of disulfonamide present to ensure the quantity is acceptable is or not. Because if there is a means increased concentration of like the disulfonamide in the body, it may result to the some toxicity. Means I would like to share that there is some drugs which have been earlier used in the treatment but nowadays obsolete from the market. We can see like various penicillin derivatives no one is using nowadays because they possess some severe side effects. So that's why the concentration or the limit should be fixed. Then inorganic impurities, so they arise from the manufacturing process and they may be of known and identified. Inorganic impurities are determined when the drug substance is tested, not in the final dosage form. It may include reagents, ligands, catalysts, heavy metals, inorganic salts. Then most of the countries used to follow their own pharmacopoeial methods for the testing of these types of impurities. Like in case of inorganic is called residue on ignition. Then we used to determine the amount of impurities of different metals. Then residual solvent and water. So we in our lab use Carl Fisher method to detect any traces amount of water. So residual solvents which are used during the manufacturing process of a drug may be detected after the product is in its final form. We use means various solvents like benzene, chloroform during like workup process or during separations, solvent extractions. So there is a means chances of presence of these means solvents in your finished product, which may comes from different stages in the manufacturing processes. It may be during active substances granulations, but nowadays granulation process already what are the various steps supposed to be followed, and what are the various ingredients supposed to be added. Everything has been prescribed or notified to the all the industries by the FDA. Then milling, then drug product coating. Nowadays, our like sugar coating, most of the companies have been not using. They are using some alternate methods. Then the most common technique for measuring the residual solvent is gas chromatography because of the small size and volatile nature of solvent molecules. External standard solution of the solvent are prepared and they compared to the sample solutions. The amount of residual solvent can then be calculated using the peak responses. Then there are some non-specific methods to determine the impurities like loss on drying, determine the amount of volatile components that are released from a sample under specific temperature and or vacuum conditions. Then TGA, that is thermal gravimetric analysis, measures the loss of volatile components from a sample over a temperature gradient. Then what are the advantages of using this TGA techniques? So this is relatively fast because these are the means manufacturer needed some kind of a speedy processes to deliver the APIs as per the demand. Then disadvantages, these are the means non-specific, we can't predict, then can't account for volatile components that are trapped in the lattice structure of the compound. So this one is a point to be noted by all the participants if you are doing the analysis by using these TGA methods. Then water. So basically water is our universal solvent, we can say. We use in most of the preparations until unless if there is some another specified solvents. So in some cases a specific amount of water is desirable, that is in hydrates. In other cases it is advantageous to keep water level to a minimum because water itself can facilitate the degradation of a drug substance. Some drug substances are hydrophilic, so knowing how the compound picks up water during stability studies is important. So then there are means various techniques which are used to determine the content of water. So that is TGA, loss on drying, gas chromatography, then dimethoxy propane titration, then near infrared NMR, Carl Fisher titration. So in industry, mostly the Carl Fisher instruments is used. Then dimethoxy propane titration. It is a thermometric endpoint is there. It is very fast and instant. So that is difficult to work with for some drug substances. So that is the only limitation with this dimethoxy propane titration. Then NMR spectroscopy. So it's a kind of a lengthy process to get the data and the sampling part. So it's not to be practical. We can be used in cross validation studies to support the primary methods. So as we all know that NMR is again further classified in the different categories. Then near IR spectroscopy, which provide good selectivity and sensitivity. A quick and non-destructive technique because your sample is there is a loss of sample, but nowadays uh, some advanced IR spectroscopy is also available. Then secondary method of analysis for water. Then call Fisher titration. So I have described in like a little bit more because most of our students is used in their laboratories. So it is introduced in 1935. So the basic reaction is water quantification in this call Fisher titration is oxidation of sulfur dioxide with iodine. So this elementary reaction consume exactly 1 molar equivalent of water versus iodine. So this is the basic principle where iodine is added to the solution until it is present in excess. marking the end point of the titration, which can be detected by the potentiometry. The reaction is run in alcohol solution containing a base which consumes the sulfur trioxide and hydroiodic acid is produced. So this one is very frequent method used very frequently for the detection of presence of water in formulation or in APIs. So when we should supposed to perform a volumetric coefficient titrations, so there are two basic classes of reagents which are very frequently used. The one component reagents contain all of the reactants like iodine, sulfur dioxide and a base in an alcohol solvent. These reagents have large water capacity and they are economical and easy to use. The sample dissolved in alcohol, but co-solvent can be used to improve sample solubility. Then two component volumetric reagents required to separate solutions. The solvent contains sulfur dioxide and base in an alcohol. The reagent is a solution of iodine in methanol with a known titer, and the main advantage is faster titration and greater accuracy for small amounts of water estimations. So there are, however, more possibilities for side reaction within a two component system which is used in Kohl-Fisher method of estimation. Then degradations. So impurities resulting from chemical change in drug substance brought during manufacturing and or storage of the new drug product by effect of light, temperature, pH, water or reaction with excipient and immediate container closure system. So this is the schematic diagram of forced degradation studies of drug substances and drug product. So if it is solid, then photolytic thermal or thermal humidity solution or suspension, acid base hydrolysis, then oxidation, then drug product if that is solid, semi-solid or solution or suspension, then we can opt this photolytic thermal or humidity oxidity analysis, then photolytic thermal and humidity or photolytic thermal oxidations. So these are the various stress conditions used for the degradation of drug substances. So degradation conditions, mainly hydrolytic conditions, oxidation, photolytic and thermal conditions. So hydrolytic condition is one of the most common degradation chemical reactions over a wide range of pH. Chemical process that includes decomposition of a chemical compound by reaction with water. Under acidic and basic condition involves acid or base stress testing involves forced degradation of a drug substance by exposure to acidic or basic conditions which generates primary degradants in the desirable range. So selection of the type and concentration of acid or base depends on the stability of the drug substances. For acid hydrolysis, hydrochloric acid or sulfuric acid, that is 0.1 to 1 molar. Then for base, we use sodium hydroxide or potassium hydroxide in the same concentration, which are suitable for the reagent for hydrolysis. If the compound for stress testing are poorly soluble in water, then co-solvents can be used to dissolve them in hydrochloric acid or sodium hydroxide. The selection of co-solvent is based on the drug substance structure. So structure of a molecule is very important for the selection of the solvent. Stress testing trial is normally started at room temperature, and if there is no degradation, then the temperature is supposed to increase. Then stress testing should not exceed more than 7 days. Then oxidation conditions. Hydrogen peroxide is widely used for oxidation of drug substances in forced degradation studies. But other oxidizing agents such as metal ions, oxygen, and radical initiators can also be used. It is reported that subjecting the solution to 0.1 to 3% hydrogen peroxide at neutral pH and room temperature for 7 days or up to a maximum 20% degradation could potentially generate relevant degradation products. Even the oxidation degradation of drug substance involves an electron transfer mechanism to form reactive anions and cations. Similarly, amines, sulfides, and phenols are susceptible to electron transfer oxidation to give anoxides, hydroxylamine, sulfones, and sulfoxide. The functional group with labile hydrogen like benzylic carbon, allylic carbon, and tertiary carbon or alpha positions with respect to heteroatom is susceptible to oxidation to form hydroperoxides, hydroxide, or ketones. Then photolytic conditions. Photostability studies are performed to generate primary degradants of drug substances by exposure to UV or fluorescent conditions. Samples of drug substance and solid or liquid drug product should be exposed to a minimum of 1.2 million lux and 200 watt per meter square light. The most commonly acceptable wavelength of light is in the range of 300 to 800 nanometer to cause the photolytic degradation. The maximum illumination recommended is 6 million lux. Light stress conditions can induce photooxidation by free radical mechanism. Functional group like carbonyls, nitroaromatic, N-oxide, alkenes, aryl chlorides, weak carbon hydrogen bond and oxygen hydrogen bonds, sulfides and polyenes are likely to introduce drug photosensitivity. Then thermal conditions, example dry heat and wet heat should be carried out at more strenuous conditions than recommended under ICHQ 1A accelerated testing conditions. Then samples of solid state drug substances and drug product should be exposed to dry and wet heat, while liquid drug products should be exposed to dry heat. Studies may be conducted at higher temperatures for a shorter period of time. Effect of temperature on thermal degradation of a substance is studied through the Arrhenius equations, that is KAEEART, where K is specific reaction rate, A is frequency factor, E is energy of activation, then R is gas constant, T is absolute temperature. Then thermal degradation study is carried out at 40 to 80 degrees Celsius. Then chemical stability of drug. Drug substances used as pharmaceutical have different molecular structure and therefore susceptible to many and variable degradation pathways. So molecular structure is very important with respect to the therapeutic or bonding to any receptor molecules, and that is directly proportional to the biological activity of any drug. So molecular structure is going to be very important when we are discussing about the impurity profiling. Then predicting the drug stability would be very helpful in design of stability study in earliest stage of drug development and identifying the way to minimize the chemical degradation. So there are processes like hydrolysis, dehydration, isomerization, and racemization, decarboxylation and elimination, oxidation, photolysis. Hydrolysis, so it is major cause of deterioration of drug, especially for those in aqueous solution. Then dehydration, removal or loss of water. Sugars such as glucose and fructose are known to undergo dehydration to form 5-hydroxy-methyl-furfural. Erythromycin is susceptible to acid catalyzed dehydration. Then we also known this isomerization and racemization. So isomerization, that is conversion of active drug into less active or inactive drugs. Then racemization, that is conversion of optically active drug into its enantiomers. The best known racemization reaction of drugs are epinephrine, pilocarpine, ergotamine, and tetracycline. Then decarboxylation and elimination. Drug substance having carboxylic acid groups undergo decarboxylation reaction. 4-amino-salicylic acid is good example of this reaction. In elimination reaction, two substituents are removed from molecule in either a one or two step mechanism. Then oxidation is also. So drug can be affected by the availability of oxygen. Some photodegradation reaction involves photooxidative mechanism that are dependent on the concentration of oxygen. Then oxygen also participates as reactant and also alters the degradation rate. Then photolysis, reactions such as oxidation reduction, ring alteration, and polymerization can be catalyzed or accelerated by exposure to sun or artificial light. Then photolytic degradation can be very complex. The products such degradation being numerous and difficult to identify. Then exposure to light can cause discoloration of both drugs and excipients even when degradation is modest and non-event detectable analytically methods. This can lead to off-color product perceived by the patient as a quality deficiency. Then physical stability of drug substance. So drug substance exist in various microscopic physical state. For example, it may be amorphous, crystalline, hydrated, and solvated state, or it may be any other form too. With time, drug or excipients change from one state to more thermodynamically stable state. Rate of conversion depends on the free energy difference between the state and energy barrier that must be overcome for the conversion to take place. So, like crystallization of amorphous drug, transitions in crystalline state, formation and growth of crystals, then vapor phase transition including sublimation, then moisture adsorption. So crystallization of amorphous drug. So as we know that poorly water soluble drugs formulate in amorphous form because the solubility of amorphous form is generally higher than of same substance in crystalline form. But crystalline state has lower free energy, so the amorphous substance tend to change their more thermodynamically stable crystalline phase with time. So therefore, crystallization of amorphous drug substance may occur during the long-term storage and may lead to change release characteristics of drug. So if there is a release of alteration in the release of the drugs, they may means alter the pharmacokinetic parameter of the drug. So like drug substances, so there is a change in like their clinical behavior or toxicological behavior of a candidate, that is drug. So as amorphous nifedipine is prepared by the spray dried method, which also exhibit the time dependent crystallization result into the altered solubility and dissolution behavior. This crystallization can be inhibited by addition of beta cyclodextrin. Means prior to this one, I would like to apprise you all that means drugs are means available in the lab, but we are unable to deliver in a desired form because of its complexity or means like in a very raw form. So when we go through our papers or the researchers, so there are numerous drugs already have been prepared or the synthesized or have been flagged, but the process of delivery to the human body is very difficult. So let's move to the transition in crystalline state. So polymorphs are different crystalline form of same drug. Because this form have different free energy and chemical potentials, depending upon temperature conditions, transition between polymorph can occur. So polymorph transitions during storage can alter properties of the drug because the solubility and dissolution rate of substance generally vary with change in the crystalline form. Temperature and humidity affect polymorph transitions. So polymorphic transition observed between two crystalline form of benzoxoprofane and two form of pyridoxal phosphate. So transition between anhydrous and hydrate form have been reported for many drug substances such as phenobarbital, nitrofurantoin, and theophylline. Significant difference in solubility can exist between anhydrous and hydrate form of same drug. So formation and growth of crystal molecules in crystal should not be considered static. Crystal can grow or decrease in size provided that there is a medium across which molecule can travel. It may liquid or gaseous phase into which molecule can sublime, like drug substance in solid dosage form such as tablet or granules may crystallize or sublime onto the surface of solid dosage form during storage. So-called whisker crystallization was observed in tablet of caffeine anhydride, as means we can say carbamazepine tablets containing stearic acid from means column shaped crystals on tablet surface during storage at high temperature. So this growth of crystals on any like a dosage forms represent its degradation or certain type of impurities over there. Then vapor phase transition including sublimation. Some pharmaceutical compounds that sublime easily may undergo change in drug content because of the sublimation of drug substance or excipients. For example, in case of nitroglycerin, which is liquid with a significant vapor pressure, so the sublingual tablet of its show significant variation in drug content during storage because the inner tablet migration through vapor phase. This transfer can be inhibited by adding water soluble non volatile fixing agents such as PEG, that is polyethylene glycol is. Then moisture adsorption, drug degradation in heterogeneous systems such as solid and semi-solid states is affected by moisture. Moisture plays important role in catalyzing chemical degradations like water participation in drug degradation process itself as reactant that may leading to the hydrolysis, hydrations or other reactions. Then here degradation rate is directly affected by the concentration of water, hydronium ion, hydroxide ion. Then water absorb onto the drug surface and form a moisture absorbed layer in which the drug is dissolved and degraded. Example sodium ampicillin, then potassium propycillin. Isolation and identification of impurities or degradations. So identification of degradation product can provide an understanding of impurity formation and define degradation mechanism of a APIs. If the identification process is performed at an early stage of drug development, there is adequate time for improvement in the drug substance process and drug product formulation to prevent these impurities and degradants long before the final filing stage. That's why the process from the procurement of raw material till finished products having more than 15 to 20 steps of checks. Then number of methods can be used for isolating these impurities or these degradants, very frequently used in our like QC department, TLC or flash chromatography, that we are use some pressure, then column chromatography, then preparative HPLC we can also use. Then the reason behind discussing all these things about this impact of impurities in active pharmaceutical ingredients, because that is going to affect our health. So these impurities can be toxic in nature. They can degrade the APIs. They can alter the quality of the APIs. They can render the API unusable. So these should be quantify the presence of impurities. They all should be by the rigorous analytical methods we can identify them, when we can separate them, and when can eliminate them from the APIs or the finished product to get the desired therapeutic effect. So all impurities needs to be identified and quantified. So all the pharmacopoeia like Indian pharmacopoeia or British pharmacopoeia or Japanese pharmacopoeia or Chinese pharmacopoeia, if you have a look on the monograph, in the end there is a list of impurities reported in the pharmacopoeia. So that's why every 5 years or 6 years, these our official books to be revised, to or the FDA or the regulators used to revise, and they keeps on increasing the list. If you visit the Indian pharmacopoeia of your 1986 or like 2014, you can see last various impurities have been included even in case of the like the CNS drugs too. Then limit has to be defined for consumption. So in this case, like a clinical trial or animal studies used to help us. If they are present, then their limits are supposed to be fixed. Then proper validated methods has to be used for quantifications. So methods nowadays we are using like AI techniques for the detections. Then genotoxic impurities has to be identified, characterized, and removed from the various processes. Then their limits. So limits of the APIs basically depend upon the uses of the drug. The drugs which are like dietary components of our daily life, drugs which belongs to a diabetic patients. So on the daily basis, they should supposed to have 2 to 3 gram of tablets on daily basis. Means thrice in a day, 21 times in a week. So you can assume for a year and year, for an year and year. So after 20 years, 30 years, the capacity of the liver for metabolizing these drugs, it get difficult. So that's why the limit of these impurities supposed to be fixed by the regulators depending upon their daily uses. Even in case of our the drugs which are used in the treatment of our brain disorders, then normally as per the our official books, it is not more than 0.1% on the API quantity. If they are genotoxic, it is not more than 50 ppm, or based on the toxicity it also can be even be less. and can be at like PPB level. Then ICH guidelines will be used for specifying the limits. The limits vary from country to country. So like US FDA for US, then for India is a then different, then for Australia is TGA, FDA is for India. So each country will have its own guidelines depending upon their environmental conditions too. Because if you are visiting like African continent or the temperate regions, it may be even Gulf countries too. So in that case, the drug usage is prescribed. This drug can be used only in India, Nepal, like this. So, so mainly we all are used to govern by this ICH guidelines, which are commonly followed for this detection and identification of these impurities. So, thank you. So, if any participants have
Thank
questions, I'll be happy to answer.
Thank you, sir. Thank you so much for delivering such a wonderful presentation among us that is on impurity profiling. I think this presentation is valuable for technical as well as non-technical persons who are using or facing the toxic materials. Now, I would request our panelists to start Q&A session. Over to Mr. Honorable Professor Dr. T.R. Bhardwaj, sir.
Hello.
Hello.
Yeah, first of all, I would like to congratulate Dr. Ravinesh Mishra for giving enlightenment lecture on regarding impurity profiling, which is one of the very important area, specifically when the impurities are of toxic level, you see, maybe parts per million or parts per billion. That much also cannot be tolerated, you see. So, in such cases, you see, specifically the impurities less than or parts per million or billion also. What is the usual approach for their quantification, you see? And how can we quantify or those kind of things?
Sir, very good question, sir. Like quantification of the limits of these impurities. What I have seen in last 10 years, the values which is showing in our like present pharmacopoeia, that one is having some variation in the previous prescribed pharmacopoeia. So I think every 5 years the limit used to get changed depending upon the data means available in the market or the post market surveillance studies. So but in case of like allopathic system, we can fix the limit of these things, but some other mode of medication system like ayurvedic, there is a they prescribe the excess doses also, which results or damage our means when that is a medication system.
Yeah, good. And second, I would like to ask, not ask, but say, as we all know that in the case of impurity profiling, the reference or the impurity which is to be again synthesized is very, very important. Synthesis of that impurity is required prior to its this thing, identification and characterizations before the approval or this by the drug rating authorities. So that is one of the area which is of concern, you see. As and when we will have, say, bulk drug parks or APIs, isn't it? So it is one of the area which is of highly importance, and there may be numerous job opportunities with respect to the synthesis, rather microsynthesis, and identification characterizations. So I think organic chemistry people or the person from the medicine chemistry, they will be of very helpful, not only in the synthesis, but their characterizations, identifications of the compounds also. Otherwise, what we have seen, even the degraded products also, with respect to the stability profiling or stability studies, if we are getting some, say, degraded product, so for its authentication, for its characterizations, to prove that it's the same degraded product, has to be synthesized and compared. And that's the reason I think that these samples, reference samples, are very, very expensive, very, very highly expensive. So what do you say about these opportunities and the synthesis or the what you call the custom synthesis of these impurities?
So rightly said by you, and focused on the main thrust area with respect to the research involved in the impurities. This is the time where we can focus on the existing drugs. We can isolate the existing impurities in a molecule and work on that. Even we can synthesize on those impurities, and we can check their biological efficacy. If they are synergistic in nature, we can develop another molecules for the treatment of diseases, like different types of the isomer of a same molecule used for the treatment of different diseases. Similarly, if they are antagonist properties, in that case we can diminish or retard the formation of these impurities in the APIs. Basically, this how we should what methods should supposed to opt for the synthesis of these impurities is very difficult, because they all are the very similar to the main component. For example, when I have done project in my master, I have synthesized impurities of amitriptyline. So amitriptyline, that is a molecule, but the impurities which one was prescribed in the pharmacopoeia of other countries, that is having some spiro compounds. So in that case, synthesizing these spiro compounds, it means 15 years earlier was very difficult. Then what kind of the animal model should supposed to use for predicting their biological efficacy? So I think there is a lot of scope with respect to the research as well as the job opportunity for our like young students. They may opt their career in this field, like impurity profiling. In South India, there is a labs like Zydus Kerala or some other groups. They are having means like a different division where they used to work on the impurity profiling. They have their own R&D. But in this area or in northern India, I haven't seen so far like any lab those who used to work on this synthesizing these impurities, their characterization, then biological efficacy evaluations. So there is a means you rightly said there is a lot of opportunity in this sector, because already we have synthesized million of molecules when we have done docking of those molecules, but we are not working on our existing molecules. We are not trying to remove their side effects or the like groups which are responsible for this one. So For example, we can see with the toxicity or the irritation of normal salicylic acid has been simply removed by the esterification of that process, that is aspirins. So like similar methods our means scientists may follow to develop a molecule which may be safe and having some lesser side effects, because it's a kind of a fashion. The drugs which we were using in our 80s, now they are obsolete. Every year the list of the banned drugs is keeps on increasing. Means in recent years, I would not like to name the drug on like open platform, that have been banned from the market. They are not usable. So I am the same person who have that nimesulide 5 years back on the prescription of the drug, but nowadays I have to supposed to have something else. So rather than banning any drug, we should supposed to check the what are the other contents or the chemical modifications we can do on that, and we can identify the in means their impurities present in such types of the drugs, and we can work on that part too, sir.
Okay, thank you. Thank you. Over to
Thank you, sir. Is there any participant who is eager to ask question from sir? Kindly raise hand. There is no one. Dear participant, due to some unavoidable reasons, our next presenter, Dr. Aleksandra, would not be able to present today. With this, we are going to invite you to join presentation at thank you. Thank you so much for being connected with us. Till then, take care. Kindly raise hand.
Every year, the list of the banned every year the list of the banned